Senators will hear from, to wrap up panel one, Dr. David Wiseman.
Never mind.
Dr. Wiseman, do you need some water, or did it?
Yeah, never mind. I'll.
I think if you press that button, it might work.
All right, I'm good. Oh, thank you very much. There we go.
Dr. Wiseman, and pull that microphone close to you. And if you would state your name for the record and who you're with, and you may begin your testimony.
Thank you. My name is Dr. David Wiseman. My company is Synekion, and I'm going to be talking about work of a group which we've operationally and informally called for the moment COVID-era Cancer Uptick Study Group, CCUSC.
Okay, great. And Dr. Wiseman, pull that microphone just a little closer and straighten it out so we can hear you good.
Do you want me to repeat that?
No, gotcha.
Okay, thank you. Thank you, Madam Chair. Thank you, members of the committee, for inviting me to participate in this important discussion. On page 2 of your slides, I just want to acknowledge my many collaborators in this coalescing collaboration of people concerned for this issue, including very highly qualified cancer researchers, epidemiology, cancer biology, the South Carolina equivalent committee to this one, React 19 of the Long COVID Foundation patient group, as well as my current collaborators. On page 3, it's a recount of some of the information that's already been presented, talking about early onset cancers, as well as a number of papers that are appearing in the peer-reviewed literature on cancer-related mortality in the COVID era. And on page 4, I'm going to show you some work of my collaborators based on UK data from their national statistics. My collaborators are quantitative macroeconomists, and they looked at the trends in England and Wales. And I'm going to show you several graphs similar to this, so I'll explain what they are so that you can easily read them. On the top panel on the right, you see trends for male and female going down, as we've heard from previous speakers, and it goes down to about 2019. Then we extrapolate the trend, and then we look at the new points in 2021 and 22, and we measure the statistical difference indicated by the green arrow there from between the trend and the new data point. On the bottom graph is the size of that trend, and all you need to focus on are the last 3 sets of bars from 2021 and 22, and anything above the dashed line is considered the statistical significance. So 2 is usually considered the threshold. You'll see that in some of these cases we go very, very well beyond that to what are considered extreme events, and this is something called the Z-score for those who know what that means. So this is the trend from the UK data, and on page 5 they continued looking at the disability claims. And just to focus on the right panel for a moment, they found a 39% increase in disability claims in the UK. And on the left panel, you can see there are 2 sort of phases to that. One is in early 2020, a spike, and then it comes back down again, and then going up again in early 2021, coinciding with the onset of vaccination indicated by the dashed line. So these are the UK numbers. On page 6, we started to look at the US numbers. This is taking CDC Wonder data, and again, very similar pattern, the way these graphs are presented, male and female. And this is looking at the early ages, 15 to 44, because of the concerns about early onset. So we certainly see that that's going on here. But this is not age adjusted for a fairly wide cohort. So on page 7 and onwards, we are now going to drill down into 10-year bands. These are the bands that's set by the CDC. And all you need to do is get a general idea of the trend. So there are 4 age bands here, going from 15 all the way to 54. And you can see in various combinations, signals occurring in the last 1 to 3 years, and above the dashed line, either male, female, or both in different years and so on. But you can see that the signals are definitely there for this mortality. And on page 8, it continues the age bands. And the unusual part is the age band 55 to 64, where we see a reverse signal. So actually the signals are going the opposite direction for that particular age band, but then resuming for the older age bands, the signals go above the dashed line, and those are very, very strong signals. So rather than talking about just early onset cancers, we at least the data that we're looking at, and these are minimum age bands, so that you can't age adjust them in the CDC output, but these are very, very strong signals. So page 9 is a sort of a summary of that, showing the age groups again on the horizontal axis. And if you just focus on the lower panel there, you can see that there's signals for all age bands except that particular one that I pointed out. So that's what we're seeing in the data. The 23 data was preliminary at the time of doing this. I think we can now do it shortly. On page 10, we asked the question, what are the potential underlying causes of this? And the first 2 bullets really echo some of the comments of the other speakers, particularly in COVID. There were delays in screening, diagnosis, treatment and reporting, the health behaviors that we've heard about. And of course, this could be a multi-hit hypothesis. But the controversial hypothesis that we're going to talk about is that we cannot exclude for the COVID era the potential of exposure to either the SARS-CoV-2 virus or the COVID-19 vaccine. And this is an unpopular comment to make, but we have to do it. We hope we're wrong, but we have to do it, and we have to raise the issue. We have to study it methodically and responsibly, and that's what we're here to request of the community. On page 11, we talk about some of the mechanisms, potential mechanisms relating to the SARS-CoV-2 virus itself. All I'm going to do is point out 2 particular bullets. The second bullet is regarding work that was actually done at UT Houston and MD Anderson, and the fourth bullet, the potential that the spike protein, those red things sticking out from the virus in the well-known picture that everyone's seen, the work of Dr. Elderi from Brown University, showing that this spike interferes with one of the main cancer protection mechanisms that we have, called P53. On page 12, we begin to talk about, well, what about the COVID vaccines? So let's go to page 13. And the first thing to note is that the COVID-19 vaccines were not tested for cancer effects, which you would expect in a new pharmaceutical, and these are documents from the package insert or FDA documents that are publicly available. On page 14, we see that there is an increasing number of case reports in the literature. A case report, again, does not prove causality, but there are a number of case reports growing of associating cancers and COVID vaccination. Page 15, we're looking at VAERS, which is the Vaccine Adverse Event Reporting System, publicly available. And the 2 main points of this slide are to note in the top left that the number of reports per year for COVID vaccines is between 18 and 33 times more than the number of cancer reports for all other vaccines for all other years combined. The big graph table on the right-hand side shows this is CDC's own analysis. This is not ours. This is CDC's own analysis that was released from a FOIA, and their analysis was performed in July 2022. The key point to note here is in the green box, if there's a number bigger than 4, and in the red box, it's a number bigger than 2. By the way, the yellow highlight is their highlighting, not mine. But any more than 4 or more than 2 in the 2 boxes, that indicates a signal. Again, the signal does not prove causality. It means check your engine. And you can see there's about 12 or 15 different cancer types for which there is a signal for the mRNA COVID vaccines. To our knowledge, this has not been addressed publicly by any of the regulatory agencies or the manufacturers. But this is something that we need to look at. Page 16. What are the plausible mechanisms why the vaccines may be doing? I again stress this is putative, and so I'm just going to address the items in black on this slide. Anything more or less anything, not everything that pertains to the virus itself could pertain to the vaccines themselves, and there's residual DNA that's in the mRNA vaccines. It's there predictably. It's needed for the production of the mRNA, so it's not unexpected that it's there, but it has consequences that I'm going to talk about in a moment. And particularly there's adducts that form between the components of the DNA and the lipids, the fats that are in these bubbles that are used to deliver these vaccines. And so I'm going to address the items in red separately now. On page 17, this very long word, N1-methyl pseudouridine. Okay, this is something that is used in these vaccines. It replaces the gene letter U in the genetic code of the mRNA, and it's there deliberately to enhance the stability of the mRNA. And this is what won the Nobel Prize for Karika and Wiseman. Without this innovation, these vaccines would not have been able to go forward. Okay, so that's good news for the vaccine itself, but there are concerns. For example, in this report, in the bottom of the slide, that there may be implications for cancer, and there are several potential mechanisms that we should be concerned about, including one called frame shifting, which I'll now address on page 18. And this is a paper from the journal Nature, prominent scientific journal, and talking about this phenomenon called frame shifting that occurs because of this methyl pseudouridine. And what's happening is that this actually makes the genetic code error prone, and it causes the production of unintended proteins because there's gene misreading. And it appears to occur in at least 25 or so % of patients. And to understand what frame shifting means, I've just given this sort of example here using words of 3 letters, which is each gene word is 3 letters: Abe, Dan, T, Tan, Ira. And if you instead of start reading that from the letter A of the word Abe, you start reading it from the letter B in groups of 3, and we come up with another sentence: bed, ant, eat, 1 rat. So you can see that by just shifting over by 1 letter, by frame shifting, we can change the meaning of this simple sort of example sentence. But this is what's happening in the biology. And this work was being done by the equivalent groups equivalent to NIH in the UK. So this is a very highly credible set of reports, and it's saying that there are unknown, uncharacterized proteins of unknown toxicology and distribution being produced by these vaccines. And in the words of the authors, this has a huge potential to be harmful. Now, they only had 21 patients who were pre-selected, essentially, and they said, well, we had no evidence of adverse events in those patients. But of course, this is a very widespread problem that needs to be addressed, and we've talked about that in a preprint shown there. On page 19, why are we concerned about DNA in the vaccines? Well, we know that it's there. That's not a surprise, but it's widely acknowledged by FDA, by Moderna, by Pfizer, by regulatory agencies, that the DNA in vaccines could be oncogenic. It means could be cancer promoting or causing. Okay, and this is widely acknowledged because DNA is present in many vaccines because of the way they're made in chicken eggs, and so on. And there are standards to control for that. But on page 20, the key to note is that those standards were written at a time before we had these things called lipid nanoparticles. Lipid nanoparticles, LNPs, are the game changer of the COVID-19 vaccines. They are what get the mRNA into the cell in rates tens or hundreds, perhaps, times more than naked DNA. And so the guidelines were written only in the naked DNA era, not in the era of lipid nanoparticles. So that's a huge problem. On page 21, we can report in works that I've been involved in that we found, as predicted, residual DNA in the vaccines which approach or exceed the guidelines. And there are some issues with how actually to measure it, and that's a concern. And in the case of the Pfizer, but not the Moderna, there are additional sequences that have no business to be there. Notably, something called an SV40 promoter enhancer sequence. And on page 22, I just want to point out that we are not talking about whole SV40 virus. We are not talking about whole SV40 genome. That is incorrect. But we are nonetheless talking about sequences which are called regulatory sequences. And there are at least two mechanisms why that could contribute or promote in some way cancers that have been published in the medical peer-reviewed literature that are shown on the right top of the panel there. On page 23, I can report that the work that we've done has been replicated in various configurations by a number of groups, and that number keeps growing. Recently, Dr. Koenig from Germany, also just to note that her estimates of the amount of DNA is actually, we think, is an overestimate to do with an artifact of measurement, which we're going to address in a future publication. But there is preliminary evidence of genomic integration. That means DNA getting into the genes in cancer cell lines. Okay, this is preliminary work. The DNA may replicate and it may mutate. And this work needs to be confirmed and extended and refined, but that's something we need to address. So on page 24, I'm summarizing the body of the work, which I'm not going to go down in detail. You've already heard my presentation. On page 25, we have to get this right for two reasons, at least two reasons. One, we know that there's been a decline in trust in public health. This has been well talked about, for example, in this article in Nature. And this decline in public health, which is having offset consequences to other types of vaccinations, has come about in the COVID vaccine era. And what we're talking about has implication for other gene therapies. And the very elegant CAR T cell therapies, which have shown enormous promise. Recently FDA have issued a warning concerning the potential for them to actually promote certain types of cancers, and for someone to take a step back and really understand it. So everything we're doing here has a consequence there. And then finally, on page 26 is a list of our recommendations and requests to the committee and to the other groups on the panel. I'm not going to go into detail, but we basically request that the state of Texas invests in research to determine associations, if any, between cancers and SARS-CoV-2 virus or COVID vaccines, and to develop appropriate methods for screening, treatment, and prevention, and to fund these efforts adequately. We think that $36 million is an initial investment we're asking for. And our request to continue detailed discussions with the members of the panel, our colleagues on the panel here, to review in detail the work that we've been doing here and to determine how we might move forward. And particularly, one particular request for MD Anderson is that we really like to get hold of their genomic sequences and tissue samples, if that could be arranged. But thank you for your attention. Thank you.
Dr. Wiseman, that was a lot. Thank you for all the panelists.